Thank you, Adam. Okay, my project started with Adam wanting to look at lower member of the Union Wash Formation strata. When you look at the USGS work on this area, the type section of the middle member is about 500 meters of strata, and the type section of the lower member is 22 meters. So when we were on our scouting mission and we went out to the Darwin Hills area, we saw this big section of lower member that did not really match the type description. So thought it was interesting. Started working on it. So what was going on in the early Triassic? It was right after the Permian-Triassic extinction event, which was the largest extinction event in geologic history. Not over 90% of genera went extinct at this time. The causes of the Permian-Triassic are still under debate. The general consensus is it was a confluence of a whole bunch of different things that caused the extinction. What they do know is that early Triassic conditions were unlike the rest of the Phanerozoic. You had greenhouse conditions with high atmospheric carbon dioxide and methane. You had anoxia in the oceans, increased alkalinity in the oceans, and hypercapnia. Hypercapnia is the oversaturation of calcium carbonate, which disrupts the ability of shell-making organisms to create their shells. So you have Isozaki in 1997 showed that the anoxic conditions actually started in the deep ocean late in the Permian, and by the end of the Permian we had a superanoxic ocean. At the start of the early Triassic, the anoxia had spread to the shallow seas and created a reef gap. So there was no metazoan reefs throughout the early Triassic and into the start of the middle Triassic. What this created in the early Triassic was anachronistic facies. Anachronistic facies are sedimentary structures that do not appear very often past the end of the Cambrian because of metazoan activity that stirs up the sediment. So you get thin-bedded limestones in shallower environments where you do not usually see them. You get flat pebble conglomerates, carbonate seafloor fans, and microbialites, including stromatolites and thrombolites. Flat pebble conglomerates form when carbonate seafloor is cemented early. A storm comes in, rips up the sediment, and forms these flat pebbles in the conglomerate. Carbonate seafloor crusts form in hypercapnic conditions. The calcium carbonate precipitates out into, in this case, vertical crystals and sometimes fan-shaped crystal formations. So the black layers here are the carbonate crusts. The anachronistic facies can be used as evidence of environmental stress. Biotic recovery in the early Triassic was once thought to be lagged compared to the rest of the Phanerozoic in terms of how fast recovery returns to normal tiering and ecosystems. Lately, though, the idea has been that there's recovery in stages. So the first one to try to define these stages is Twitchett et al. In 2004. Stage 1, you have small body size, you have low diversity and high dominance, and you have low tiering in both infaunal and epifaunal populations. Infaunal communities are also very small and simple burrowing. This goes up in diversity as you get to stage 2, and stage 3 up in size. By stage 3, you start seeing higher tier organisms such as crinoids and higher tier and higher oxygen-needing animals such as crustaceans. And stage 4 is the return to a modern ecosystem with modern sizes and modern tiering. So biotic recovery across the globe was patchy and somewhat dependent on where you were on Earth. So the harsh conditions were a little more worse around the equator, so you had faster recovery in Western Canada and Oman, which are further from the equator, between 20 and 30 degrees, rather than the Western US and Northern Italy, which were closer to the equator. So the stages took a lot longer in the Western US and Italy. However, some areas like Oman, you had recovery, and then the recovery disappears. The no data they put here in their chart just means that they found no fossil evidence past the end of the Griesbachian in that area. Chen and Benton in 2012 tried to improve on this model by making a pyramid. In their chart up here, they show a full Permian reef system with top predators that was wiped out at the end of the Permian. The early Triassic is dominated by primary producers, which are microbial communities. The next things that move in are primary consumers, disaster taxa, that are usually the first generalists to move into an area that has been disrupted. Next come mesoconsumers, and this started at the beginning of infaunal tiering in the next section. Mesoconsumers 2 and reef builders move in next. And finally, you see the return of top predators, first top invertebrate predators for P1, and then you have a return to modern ecological conditions with the return of vertebrate predators. Also going on in the early Triassic, you had habitable zones that acted as refuges for different populations of animals. Along the coasts, where you had a stratified ocean, wave action would aerate the water. So you had a small zone between the fair weather wave base and the storm weather wave base, where oxygen conditions made life possible, but were bounded by wave action, which would destroy any trace makers. And below storm weather wave base, the anoxic or eugenic conditions would stifle any trace makers. Biotic recovery was mainly dependent on oxygen. So as oxygen leaves the system, you get lower diversity and lower complexity of your trace makers over time. This is also true for the other environmental stressful situations, hypercapnia, high salinity, high alkalinity. So my hypothesis was the presence or absence of harsh environmental conditions is the limiting factor in biotic recovery after the Permian-Triassic extinction event. In the Darwin Hills, however, there's extensive bioturbation to the lower member, including about the majority of the 150 meters of the lower member of the Union Wash. So what conditions would prove this hypothesis? If the trace fossils were increasing in intensity, which was measured by Ichnofabric, size, measured by Burrow Width, diversity, which I measured by the number of species present. If all those conditions are met, That means harsh conditions are absent in this particular locality, and average Phanerozoic recovery is occurring. Our location was in Darwin Hills, California, along the 190 between Owens and Panamint Valley. The black blobs here are Triassic rock. It's faulted throughout. Everything in here is Permian and Triassic. We had two localities. Burrow Hill was the lower member locality. Darwin Wash was the middle member locality that we used to compare environmental conditions post what was going on in the lower member. Union Wash was located on the western edge of the North American portion of Pangaea during this time, within 10 degrees of the equator. The lower member was in the Smithian stage of the early Triassic. The middle member bounded the Smithian and Spathian stages. The Triassic rocks can be correlated throughout the western U.S. to the Moenkopi Formation in southwest Nevada, the Dinwiddie Formation and Thames Formation in the Rocky Mountain States, and to the Vegas Siltstone in western Alberta. The geologic setting at this time, the lower member was in a shallow subtidal setting. The middle member, which is represented in the map here, you can see Darwin Hills, is a deeper environment in the outer shelf. The methods I used for this project, most of it was field work. I did field observation, did stratigraphic sections of both of my localities, did sample collecting every 1 to 3 meters depending on if the units were covered or not, and I did gamma ray spectrometry every meter or at every lithological change. The lab work, I slabbed my samples and had thin sections made. For the paleontology, did field observations, identifying any trace fossils I saw, measuring the trace fossils in 2 ways, or in 3 ways. Ichnofabric on the top and sides of the bedding, and I measured the width of the burrows. I also noted any body fossils I found and any microfossils in the thin sections. For Ichnofabric, I used the methods of Drozier in 1986 for the side of the beds and Miller and Smale in 1997 for the top of the beds. Ichnofabric measurement is basically a number given to the amount of bioturbation in the rock. So 1 is considered no bioturbation whatsoever, and 5s are considered total disruption of the bedding plane. There is also a 6 in the Drozier scale that is almost to the point of a massive look in the rock. So how does this apply in the field? If we find a site of bedding like this, you put the scale up next to it and compare to see what numbers you get. So this would be an example of a 5 throughout most of this section. For burrow width, I randomly chose bedding planes and measured 20 by 20 centimeter plots on them. And going clockwise around the plot, I measured every burrow I could find up to 50, which seemed to be right around the limit of how many burrows I could find on each of these plots. I also did gamma ray spectrometry. First, measure the thorium-uranium ratio. Uranium isotopes are stable in both oxic and anoxic conditions. Uranium, however, is controlled by the oxygen level. When there are anoxic conditions, uranium is precipitated out into the rock, So that would give an overall higher score of uranium in anoxic conditions. So a low thorium-uranium ratio indicates anoxic conditions, and a high thorium-uranium ratio indicates lower oxic conditions. I also took potassium readings, which would track changes in the classic input in the section. So what were my results? The lower member was all chalcocyltite. There were abundant trace fossils, including these big sections, and 3 out of the 4 of the blue subunits here. I divided the lower member into 8 subunits. Subunit 3 was pretty unique, so we'll go over that one. Subunit 1, it's all chalcocyltite. It's cross-bedding and channelized in places. You can see a big channel right here next to Adam. And it had abundant thalassinoides fossils. Subunits 2 and 4 through 7 were all chalcocyltite. They were also abundant thalassinoides fossils. Here we have a picture of subunit 2. Here we have subunit 5 in the foreground. This gray outcrop is subunit 6, and the rest of the top of the hill is subunit 7. For subunit 3, it was a bit different than the rest of the section. You see anachronistic facies such as flat Pebble conglomerate on the bottom, thin-bedded limestone throughout much of the rest of the section, and the presence of ammonoids. So here you have the flat Pebble conglomerate that is the base of subunit 3. We have a thin-bedded limestone with ammonoids. The little black blobs in there are the ammonoids. And the top subunit is subunit 8, and that is laminated chalcocyltite with no fossil evidence whatsoever. No trace fossils, no macro or microfossils in that one. For the middle member, it's mostly anachronistic facies. We have a lot of laminated chalcocyltite, flat Pebble conglomerates, black calcite cements, and concretions in some of the layers, and no fossils, no trace or macro or microfossils in this section. So subunit, it was divided into 4 separate subunits. Subunit 1 was all laminated chalcocyltite with limestone concretions in here. Subunit 2 consisted of interbedded flat Pebble conglomerate, black calcite crusts, and laminated chalcocyltite. Subunit 2 also showed slope features such as a debris flow right here. Subunit 3 had the flat Pebble conglomerate and laminated chalcocyltite, but did not have the black calcite crusts of subunit 2. Subunit 4, we're back to the laminated chalcocyltite with limestone concretions. In subunit 4, the concretions were often formed around rip-up clasts. You can see a little tiny clast in the middle of this concretion on the bottom here. For the trace fossils, we only found 1 trace fossil throughout the whole section, and after some digging around, figured out it was Thalassinoides suevicus. Thalassinoides have a complex system, which means there's lots of branching and there's lots of overlapping. There are smooth walls, and Thalassinoides suevicus is differentiated from the other thalassinoides in that it's a mostly horizontal gallery, and it has Y-shaped branching rather than T-shaped branching you see in the more 3D Thalassinoides paradoxicus. So for subunits 1, 4, and 6, the ichnofabric varied from 1 to 5. You can see the ichnofabric side is the first chart here. The ichnofabric top is the second chart. So 1, 3, and 5, it varied from 1 to 5. In the bottom, it somewhat builds up. In subunits 4 and 6, however, it was more varied. Subunits 2, 3, and 5, the Ichnofabric, with one little exception, and at the top of subunit 5, it was high between 3 and 5 the entire way. And in subunits 3 and 8, we saw no Ichnofabric whatsoever. For the burrow size, they remain steady throughout the section at around 3 millimeters. Small size for thalassinoides is considered less than 5 millimeters. For our gamma ray results, we are actually right below the anoxic-oxic line most of the way through the section with the exception of a couple spikes. The K percentage was slightly higher at the bioturbated units, but not significantly so. For the middle member, we actually had oxic conditions right around the line. Which makes somewhat sense because when the calcium carbonate cements are precipitated, you have oxygenated waters mixing with a hypercapnic environment. And the K percentage is higher in the units that form concretions, but is not really significantly different throughout the section. So what does this tell us about the depositional environments? The lower member was a subtidal environment. There were most likely anoxic incursions at subunits 3 and 8, as shown by the anachronistic facies that are there in those two units. And this was most likely a habitable zone, since we had our thorium-uranium ratios right below the anoxic line, coupled with the fact that there are high oxygen trace fossils in this area. It's most likely showing fluctuating oxygen conditions that the gamma ray spectrometry can't pick up. So we're most likely at the lower edge of a habitable zone. In the middle member was a deeper shelf environment with anoxic and hypercapnic conditions, as shown by the presence of the black calcite crusts and flat pebble conglomerate. So back to my hypothesis. The presence or absence of harsh environmental conditions is the limiting factor. If the trace fossils increase in intensity, size, and diversity, that shows there is recovery occurring. So what do we have? We have one genera, which is recovery stage 1. However, that one genera is a higher tier, higher oxygen needing thalassinoides, which would put us in recovery stage 3. The high ichnofabric index would put us in recovery stage 3. The small size, however, and the fact that it does not increase puts us in stage 1. So we sort of have a mixed bag of things going on here. The high ichnofabric index and thalassinoides could indicate a healthy environment. The low diversity and no increase in size and our low thorium-uranium readings could indicate that limiting conditions are occurring. There is also evidence of anoxic incursions in subunits 3 and 8, which would indicate that harsh conditions are in the region and invading this locality at different periods. So in conclusion, even though there's a high ichnofabric index, there is the low diversity and the small size, the static small size, indicate that recovery is actually limited in this area. Harsh environmental conditions are therefore a limiting factor in the recovery of this in this area. In the lower member, there was limited recovery. It was most likely at the edge of a habitable zone. And evidence of harsh conditions, there were evidence of harsh conditions intruding into this shallow environment periodically. The middle member was the return of, was the long-term establishment of these harsh conditions and the squelching of any recovery that had occurred in the lower member. So I'd like to say thanks to my advisor, Dr. Woods, for his patience and help as I took a long time to do this project. My committee, Nicole and Matt, for their help and encouragement. Thank you to my field assistants, Mike Ha, Nancy Akita, and John Sponger. My family and friends for all their support. And the CSUF Geology Department. I've actually had a lot of teachers here at this school. I was in Phil's first class to show you how long I've been here. And everyone's been very knowledgeable and encouraging the whole time. So thank you very much.